In the last quarter, quarter 3, we spent all of our time on the central dogma: replication, transcription, translation. There's more. There's actually a lot more, and that more I've always wanted to cover in this class, but we've never managed to get our act together or find the time to do it until today. So you're going to watch an N of 1 lecture, which is probably going to involve me using my notes a bit more than ordinary, probably messing up a couple of slides, and also probably you all throwing me off with some questions along the way, but that's okay. That's why we'll make it better for the next time. So be patient with me. I have no idea how long this lecture will take. I'm pretty sure we'll be okay, but just if it seems a little bit off, this is what happens when you teach a lecture for the first time. All of these things that we're going to try to cover today, we have cutely grouped as extra dogma, because they're the things that don't rewrite the central dogma, they elaborate upon it, and we'll show that schematically. And I think Professor Zunder's pre-lecture that everyone watched gave a good introduction on some of the things we'll cover topically today. Our disease wrapper for today are 2 syndromes, one which you've heard about, Down syndrome, and another one you probably have not, Klinefelter syndrome. Let's begin with Down syndrome. This, I'm sure this has touched many of you, perhaps in your family, a neighbor, relative. Genetic disorder, of which it's very clear, different developmental delays and physical disabilities. The symptoms overall, the phenotypes of Down syndrome individuals are many and obvious. I have a couple of clinical terms here. Brachycephaly is a shorter, fatter, stubbier head, cephaly head. Brachydactyly, shorter, stubbier fingers. All right, we did dactylitis, you remember, for the sickle cell anemia lecture, same roots. Duodenal atresia, the duodenum is part of your GI tract. It's supposed to be a tube. When the tube doesn't properly hollow during development, it's closed and blocked. That's what atresia is. So the blockage, developmental blockage of a tube that ordinarily should have an orifice. Epicanthal folds. I don't know if there's any Asians in the audience, but your eye folds here, that's another characteristic of Down syndrome patients. I don't have any folds in my eyes. Clinodactyly is bent fingers on the 5th finger, which I presume would be this one. And then some other things. Hypotonia, it's sometimes called, this is often an early sign of Down's patients, so-called floppy baby syndrome, so low muscle tone, so not as rigid as a typical infant would be. That propagates to ligaments. I think we're all aware of the mental impairments, shortness, and then there's some other developmental patterning defects that are listed here for completeness. The upshot is that Down syndrome have a lot of things that are out of the normal range of human variability as a result of the genetic disorder. And I want to contrast that with Klinefelter syndrome. Klinefelter syndrome is estimated to be the most common cause of male, genetic cause of male infertility. But up to 65% of the cases can go undiagnosed. Thank you. The reason why is because the symptoms of individuals with Klinefelter syndrome, it's all manifestly males, they're very subtle. Gynecomastia is development of male breast tissue, so enlarged breast tissue. Hypogonadism are underdeveloped sex glands. And associated with this are elevated levels of a hormone called follicle-stimulating hormone that's released by your pituitary gland and is important for proper spermatogenesis as well as oogenesis, the development of eggs in females and sperms in males. And it's the genetic testing of infertility, males that go in for infertility, that more often than not is what reveals that those individuals have Klinefelter syndrome. Those are the differences in the phenotype. So extensive with Down syndrome individuals, rather subtle with Klinefelter syndrome. But there are many other things that are very similar between these two. Prevalence and incidence, you've covered these terms already with Professor Barker. They're very similar. 250,000 individuals in the US. Note it's men only for Klinefelter syndrome. Incidence is about the same, so very comparable. They both appear to occur randomly. These are not inherited germline defects. We want to talk about genetic defects. And in addition, there haven't been any strong linkages with environment or behavioral activity that impacts those incidence rates. The one exception is age, that there is an age associated association with the incidence of Down syndrome and age of the mother. And there are also some anecdotal observations of increases in incidence with age of the father for Klinefelter syndrome. And we can speculate on why that might be. I'll try to give as much of the mechanisms as we understand for this class. So what is the etiology of these genetic diseases? Again, more commonalities. They're both caused by defective meiosis. We will have a whole lecture and pre-lecture on mitosis. Meiosis is a form of cell division that is specific to sex glands, sex organs, and for mating. So we're ordinarily diploid organisms. During meiosis, those 2N, the 2 copies of each one of those alleles, get split into haploid cells, 1N each. And each one of those 1N alleles has combinations of segments of the original 2N diploid germ cell that gave rise to those meiotic cells. I'm not going to give any more details on that because you could do a whole lecture on meiosis, but it involves chromosome segregation. There can be defects in chromosome segregation that can give rise to abnormal chromosome numbers, and abnormal chromosome numbers generally is called aneuploidy. Aneuploidy for a haploid cell, that should ordinarily be 1N, is 2N in this case for some specific chromosomes. When we talk about cancer, cancer can also be aneuploid. This is near the end of the class. Those are diploid cells ordinarily, so they would have 2N plus or 2 plus N genomes in them, both examples of aneuploidy. And the chromosome that is aneuploid for Down syndrome and Klinefelter syndrome are well known and are different. Down syndrome is an extra chromosome 21. All right, so what happens is that the age association of the mother, if there's an extra chromosome 21 in the egg and then is fertilized by a sperm, now you have 3 copies. So 2N plus 1N make 3N of chromosome 21. Whereas for Klinefelter syndrome, the extra copy, their thinking is it's associated with the sperm, is the X chromosome, an extra X chromosome. Both of these aneuploidy events are going to have the potential to cause an elevation in gene dosage. Gene dosage refers to the number of copies of a gene, and then by extension, the number of copies of every gene product that is encoded in that gene. At a first pass, what occurs, let's say for a Down syndrome patient, you have an extra chromosome 21. Now you have 50% more of every gene on chromosome 21. To a first approximation, you have now 50% more RNA, 50% more of all of those proteins, and this causes changes in the regulatory state of the cell, all of the transcriptional regulation that we talked about before. Everything has been disrupted by that 50% change along all the genes in the chromosome. But at the surface, then that doesn't explain why you would have one syndrome that has a pretty extensive phenotype and another that's rather mild. Why? I'll tell you what it's not, would be an explanation. Oh, maybe there's just more genes on chromosome 21 compared to the X chromosome. So it's just a numbers game. In fact, it's the opposite. Chromosome X is roughly 3 times the size and encodes over 3-fold more genes, all of which have the potential to be increased by 50%. So you can't just make a numbers argument like that. It's something else, and that's what we're going to get to today. We have 2 things that we want to cover today in the extra dogma. We'll begin with the epigenetics that Professor Zunder kicked off in the pre-lecture. if we go back to our DNA makes RNA makes protein central dogma, epigenetics impinges right at that interface between DNA and RNA. And he already gave a nice introduction of the different ways in which epigenetics can be defined. I'm not going to repeat it here today. Some people will take it as a catch-all for anything at this histone nucleosome level of regulation or DNA modifications. I'll speak more about that today. I just ask you to be aware of these differences and be careful that it's a loaded term for some people, and the older they are, the more likely it is to be a loaded term. Yes? How can you have heritable differences if they don't alter the genome sequence itself? The classic example of that, the slides are merging together to me. The pre-lecture did cover DNA methylation. So a cytosine methylation doesn't change the cytosine, all right, and that they get reset and then put back on, and so it's still a C there, and it could be a methylated cytosine or an unmethylated cytosine, and so that's the classic example. The rest of the histone modifications that we'll talk about in greater detail today, those are the ones that start to extend the line. Next couple of slides, I'm going to talk about almost exactly that, this idea of stable versus metastable epigenetic states. And there's a couple of very nice and clear examples. One is the so-called agouti mouse. This is a different mouse species that will have a yellow coat color, but the extent of the yellow versus brown is a function of the epigenetic state of a particular locus that encodes a pigmentation gene that is reflected in the coat of the animal. These are all siblings, all right? And depending upon the extent of epigenetic modifications of the agouti gene locus, you get this whole spectrum from a normal-looking mouse coat to this agouti color. And what's really interesting, this mouse model has been an exemplar for studies of epigenetics going back a long time, because interestingly, the environment can impinge upon the epigenetic modifications that are then reflected in the heritable, the coat color of the mouse. And so the diet of the mothers will impact the extent of the yellow color or the brown color in the progeny. That's a function of the diet and not the genetic makeup. There are also some really remarkable examples in humans. During World War II, Nazi Germany cut off the food supply to the Dutch. There was a hunger winter. There was a really bad famine when many people lost their lives from hunger. And people have done population studies tracking children that were in the womb during that winter and found that the stress during development appeared to put permanent marks on those individuals' genomes that now, decades and decades later, led to increased incidences of obesity, shorter lifespan, 10% changes in some of these compared to other individuals born at around the same time. And so these things are very real and can be extremely long-lasting. They can also be really short-lived. Yeah, right. Your question is how do you separate the individuals that survived during that stressful time compared to others that did not? Yeah, this is a lot of the challenges in this study. There's actually a whole suite of literature on this, trying to follow these individuals with closer and closer fidelity and things. You always have this problem of covariation. So what other things could be happening concurrently, right? Like maybe that just happened to occur. What they were largely looking at were epigenetic differences between the mom and the progeny. So to try to control for the idea of that there's an inheritance of locus and other types of things. And so it's the analyses are usually trying to balance out or control for those other covariates, and then say, after you take away all those other possible combinations, is there still a significant effect of them. That said, it's not set in stone. It's just some provocative things, and I think it's not, some people have alternate explanations for that. The agouti mouse, though, is pretty clear. Long-lived epigenetics. Let's touch a little bit on shorter-lived epigenetics. There are epigenetic modifications. Here now, we'll talk about timescales in a moment, but this reflects more of these nucleosome modifications, histone modifications. There are modifications on core histones that will go up and go down with circadian rhythms. All right, so our natural oscillations that occur in all of us because of exposure to the sun, gravitational pulls, all that type of stuff, these will change in light and during the day and at night. There are also clear examples of long-lived things, but regulated things that can get put on very rapidly and also taken away in memory, learning and memory. The cartoon example here shows one way in which it could happen. We discussed transcriptional regulation. CFOS is a mammalian transcription factor, important in a lot of different settings, but also in neuronal activation and maturation. If you have a particular event that opens up a locus, moves the nucleosomes out of the way there, and then as a result of that opening and the signals that gave rise to CFOS being able to go in and regulate transcription, if there are additional epigenetic events that modify the DNA or the nucleosomes in the vicinity to keep that locus open for longer than when the stimulus persisted, here now you can have an epigenetic state that was turned on very rapidly, but then persisted for a longer time, although we'll talk about how some of these marks can get reset. We forget things, okay? Things get reset. So there's another example of some reversibility in epigenetics. Structured this lecture on two main categories of epigenetic modifications, epigenetic mechanisms that relate to what Professor Zunder said in the pre-lecture on chromatin accessibility. DNA methylation, more to come on that in a slide. This is a reversible modification at the DNA level where the relationship is pretty clear. If you have methylated DNA, those will promote compaction of the chromatin, and that is a mark that will repress or silence gene expression, transcription in that locus. One that's a bit more complicated are histone modifications, because the nature of the modifications can be dramatically different. The impact of those modifications on the local chromatin environment depends upon the modification itself. And thus there's a higher degree of versatility, and there's also a pretty strong separation in time scales about how long lived any of those histone modifications are on any given histone or nucleosome. So we'll begin by speaking more about DNA methylation. The pre-lecture introduced this in very broad, high-level terms. I want to talk about a bit more mechanistic detail about how it's regulated, because some of these developments are relatively recent. Here's a schematic of the pathway for DNA methylation and the path to demethylate a methylated cytosine. Let's orient you. We begin with a regular cytosine. This will be a cytosine in one of those CPG islands or motifs. It is modified to a methyl cytosine via an enzyme-catalyzed reaction. The enzyme that catalyzes it is called DNMT, or stands for DNA methyltransferase. Hopefully that's pretty self-explanatory. The additional thing I'd like you to know is, where does the methyl group come from? The methyl group comes from a modified form of methionine called SAM, or S-adenosylmethionine. And SAM is a pervasive methyl donor in cells. You'll see it again when we talk about other methylation events. And so you need SAM as one substrate, the cytosine as the other, catalyzed by DNA methyltransferase to get you the methyl cytosine. And for a long, long, long time, that was it for DNA methylation. You put the mark on, and it doesn't come off. That was the dogma. It was only maybe 15 years ago that the first demethylation mechanisms were uncovered. And partly the reason is that you can't, the methylation mark on the cytosine is extremely stable. It's not simply a matter of just clipping off the methyl and you go back to the cytosine. And so the way that demethylation is catalyzed is by a dioxygenase called TET. The TET acronym, if I tell you it stands for 10-11 translocation, doesn't mean anything. But the TET enzyme is a dioxygenase that moves the methyl cytosine to a progressively oxygenated state that will be recognized by the base excision repair machinery. Recall those enzymes that will recognize a DNA lesion, come in, cut it out, and fill in, ligase, all that stuff from DNA repair. This pathway exploits that repair mechanism to go back to the original cytosine. So all of these different oxidized or oxygenated states of the methyl cytosine can either be passively demethylated just by virtue of their modification, or they'll get recognized by the base excision repair machinery to go back to the original cytosine. So it's cut out and then put back in by a polymerase. As was said in the pre-lecture, these methylated cytosines reside in clusters of cytosine phosphate guanine bases, or so-called CPG islands. These are the ones that have the potential to be methylated. This is also the reason why unmethylated CPG, if I can circle back to PAMPs and PRRs, you remember CPG as a PAMP for toll-like receptors. This is the reason why most of our CPG islands in the genome are methylated. It's by virtue of this, these enzymes and the modification on these loci. When you have a CPG island that's methylated, you'll often see these CPG islands upstream of genes that need to get shut down in certain differentiated cell contexts. And so these islands, when they're heavily methylated, will shut down the ability for gene transcription in the vicinity of those islands. DNA methylation also has some other interesting characteristics. One, we're talking a little bit about development, meiosis and things in the beginning. The one place where methylation is known to be erased completely is during formation of the zygotes. All right, so during meiosis and the formation of egg and sperm is when you wipe clean all of the methylation events and then reset them afterwards. The erasing and resetting and the idea that the methylation marks are going to be an indication of the egg or sperm from which the allele came from is the basis for this phenomenon called genomic imprinting, which I'm going to talk about in a cartoon form because it's a little bit complicated. Here's my cartoon. We're going to begin on the left with what are called the primordial germ cells. These are the progenitor cells that eventually give rise to egg and sperm and females and male. It's these primordial germ cells. Oh, and I also have to orient you because this cartoon looks nice, but it's actually really complicated. Pink indicates maternal alleles and a maternal environment. So female allele in an egg, female allele from the mom of this male in a male primordial germ cell. And then vice versa, you have the paternal allele in either a maternal environment or a paternal environment. During that process of meiosis, where you go and you segregate the gametes, things, all of the marks that would be specific for either the male or the female are erased. Oh, and the other thing I should say is that those methylation marks here are in the black and in the white. So black is methylated, white is unmethylated. Circling back to the same coloring scheme like shown here. So during oogenesis or spermatogenesis, you wipe all those methylation marks clean, segregate the individual haploid cells over here, and then as that segregation occurs, the methylation marks are established that are specific to the environment in which those gametes exist. And so in the female now you have two different haplotype cells, each with the maternal environment, here each with the paternal environment. During fertilization, now you have a for each chromosome, you have, by the methylation marks, a chromosome from your mom and a chromosome from your dad. And then that resets down from here. Who cares all about this complicated description? Well, if there are differences in the methylation pattern between your mom and your dad, now those genes on each one of those chromosomes carry a record of whether or not they came from the mom or they came from your dad. And if those impact the gene expression of each one of those, that will give you differential expression of each one of the genes off of one of your alleles, either from your mom or your dad, or the other. There was a question in the back.
When the dashed lines turn from white to blue, is that just indicating that now the methylation pattern is that of the male host, or is it something else that's also occurring that's like adding on the blue or the pink?
Yes, your question is, there, here, the black went white, the white stayed white, and then the pink and the blue also went white. Right. Yeah, at that point, what this schematic is referring to is that because of the pattern of the methylation events, it's a maternal methylation pattern or a paternal methylation pattern. You take all the methylation events away, then you can't tell because there's nothing there to distinguish the two based on the epigenetic state of it. Then as the egg or sperm matures and fully differentiates, now those marks get put back on as a function of either being from the egg or the sperm. First, this seems rather bizarre, and then there's also like, why would this occur developmentally and things that you have to give some sort of evolutionary argument for why you might have this phenomenon of imprinting either from the mother or the father. And so there is, in fact, a theory that is a theory, has not been proven, called the kinship theory, where there are examples of alleles that are imprinted on either the paternal allele or the maternal allele, and examples of cross-communication is also there. I should mention that genomic imprinted genes, you can list, they're in the order of hundreds of genes. So we have 20,000 genes in our genome. So this is a small fraction that are imprinted either on the mom or the dad, but they're recurrent and consistent. And some of them, the examples are pretty interesting. And so one is a gene and receptor in a family that you've already heard about. Remember, anabolic steroids and IGF is one of the androgen receptor target genes. Another family member in the insulin-like growth factor is IGF-2, and one of its receptors are imprinted genes, classic examples of imprinted genes. IGF-2, if there are individuals that have a lot of IGF-2, that an embryo is expressing a lot of IGF-2, that embryo is going to grow, the child will grow to a larger size. IGF-2 receptor is like a decoy receptor, a sink for IGF, so it's like an inhibitor almost for IGF-2 with regard to signaling and its ability to promote growth. So the IGF-2 ligand is imprinted on the dad's allele, whereas the IGF-2 receptor is maternally imprinted. And you think, why is that? Well, from the male's perspective, it's totally the selfish gene, right? If you fertilize someone, you want your kids to grow as much as possible because you've already passed on your genetic information. From the mother's perspective, thinking this is this kinship from a selfish perspective, for her to bear children, she simply wants to be able to bear as many children as possible. And so the less taxing any one of those children is, the more effective that her germline will get passed on to more kids. So it's a theory, but this is what some of the thinking might be, why there may be evolutionary pressure to have some of these imprinted genes that go along with either the mom or the dad. It's a little crazy, but that's what people think. All right, I'm going to shift to histone modifications. Some of these things professors covered in the pre-lecture. I think I only have a handful of things to add. I want to orient you again around histones themselves, very basic proteins that self-assemble with one another into an octamer of these histones 2A, 2B, 3, and 4 times 2 that come together to form the octamer. And all of the modifications that we'll be talking about in the forthcoming slides relate to these histone tails. So the N-terminal tail is not in that octamer core, and it has a number of amino acids on it that can be modified post-translationally. One of the modifications is acetylation on lysines. Lysine acetylation changes the charge from a positive charge to neutral, which will create more of will eliminate the attraction between those histone tails and the rest of the DNA that would ordinarily cause condensation of those nucleosomes. And so that acetylation event is catalyzed by HATs, or histone acetyl transferases. And then the reverse reaction, the deacetylation, is by HDACs, or histone deacetylases. So acetylases, deacetylases, acetylases open the chromatin, deacetylases close it. And the last thing I wanted to expand a bit on over the pre-lecture is why is H3K9AC in blue. I do want you to be familiar with this shorthand notation because that recurs for every histone modification that I'll show in the next couple of slides. H number, that's the histone that gets modified. K will be the one-letter abbreviation for the site that gets modified on the histone tail. Therefore, I think you will need to know that K stands for lysine. And then whatever the shorthand and abbreviation is after that is the modification. So AC is acetylation. I'll show a couple other examples, I believe, in the next slide. There are more marks than histone acetylation, and this will be the slide that goes through a couple of them. And what I've highlighted here is give you a sense of what the time frame is of these. Histone acetylation is really rapid. Okay, this accompanies gene regulation itself. All right, so histone acetyltransferases are recruited to loci along with RNA polymerases and things on that time scale. So this can happen very fast and it can get shut down very fast. So associate that with minute time scale. So T squiggly is a rough time scale that these can go on and go off. Histones can also be phosphorylated. There are, especially on serines, and this is kinases that will catalyze those phosphorylation events. So here's histone H3 phosphorylated on serine 10. The last one, which is more complicated, I think the other thing I should expand upon is phosphorylation, like acetylation, is generally thought to be active. Recall that phosphates put negative charges onto the protein. So eliminating a positive charge with acetylation or adding a negative charge by phosphorylation is the same net result with regard to opening the chromatin. Histone methylation is more complicated. And to be totally honest, I can never keep these things straight. I've been on hiring committees for epigenetic people, and I've asked them, like, how do you keep track of these things? So I'm not going to ask you to memorize these because that's a trivial exercise to go in and do. But what I do want you to know is what the shorthand is, and I want you to know the time scale. So time scale, this is the longer epigenetic modification, days or longer. And I want you to be aware structurally of what's going on here. These are also lysines that get methylated. The methyl donors, I discovered this last minute, ADOMET is another name for SAM. It's another S-adenosylmethionine substrate catalyzed event. The enzyme itself is a histone methyltransferase. All these enzymes are pretty self-explanatory, as is the reverse reaction, which here you have dedicated histone demethylases that will reverse modifications. So you can go to the right or you can go back to the left. And you can go progressively to higher and higher levels of methylation up until all of the hydrogens on that primary amine have been methylated. The charge doesn't change. but it should be clear that the overall steric bulkiness of that lysine has changed. But these more lysines out on the side, that's going to impact the flexibility of that residue, and by extension, the flexibility of the histone tail. Thus, the exact histone and site that is modified and how modified it is associates it with active or inactive states of the chromatin, which is the part I always get turned around. But what I want to emphasize is here you'd say like H3K4 dimethyl or trimethyl, so Me2 or Me3. Me2 would be this state here. Me3 is this state here. And you can have different lysines on different histones getting methylated to different extents, and those associating with active or inactive states of the chromatin in which they're found. Questions on that? If you put all of those different post-translational modifications on the histones, it gets very complicated very quickly. This term Histone Code is meant to refer to that combinatorics of what is the epigenetic landscape of a gene when you have all of the different combinations of modifications occurring at histones, nucleosomes, the local DNA environment, and then higher order things from there. Let's take a cartoon on the right. You can't see anything along these, but these are the 4 core histones, H2A, H2B, H3, and H4. This is the histone tail running along through, and all the decorations on the top and the bottom are methylation sites, acetylation sites, phosphorylation sites. If you take each one of those and say, yes, you're modified, or no, you're not modified, and take them all as independent, and then you take into consideration that there are 4 histones times 2 for each nucleosome, I think Eli did the math on this. All the combinatorics are more than the elementary particles in the universe, if you take all the combination. That's for 1 nucleosome. So there's a rich complexity in the information that's encoded here, and this is still a really active area of research that has not been fully worked out, and it's really because of the combinatorial complexity. Questions on epigenetics? All right. Second part, I want to move on to another area which wasn't touched on at all in the pre-lecture, and this is in the area of non-coding RNA. Non-coding RNA is everywhere, and we're not going to be able to do it complete justice, but we're going to get as far as we can today. Fortunately, the definition of what a non-coding RNA is pretty straightforward. Regions of the genome that are transcribed but are not translated. Any of those in principle are not, or any of those by definition are non-coding RNAs. We spent a lot of lecture time on the regulation of mRNA, genes that are transcribed. I show this pie chart here to get across the idea that mRNA is a surprisingly small amount, either by mass or by number, of the total number of RNAs that a cell expresses. We covered a lot of these other ones, transfer RNA, ribosomal RNAs, you can see them indicated here. What I want to do today is talk about just 2 other non-coding RNAs because they are important and very widespread in biology, the so-called MicroRNAs and long non-coding RNAs. They are functional even though they're not coding, akin to what ribosomal RNAs, tRNAs. They don't code for protein, but they serve important biological functions that we've already covered. MicroRNAs are important for what we'll call post-transcriptional regulation. MicroRNAs can alter the abundance, downregulating the abundance of mRNA transcripts through a process called RNA interference, or another mechanism that I'll talk about in a moment. But RNAi and RNA interference generally is something that we wanted to have you be aware of because we use this in the lab all the time. There was already a Nobel Prize given on this. It was a major discovery 15, 20 years ago. Long non-coding RNAs or link RNAs we'll give a formal definition of what they are, but they're long pieces of non-translated RNA that can serve a number of different functions. One of them is they can act as what's called a sponge, meaning complementary sequence that can bind to other RNAs in the cell and act as like a sink for them to prevent them from executing their normal function. Also, many of them are so large that they'll adopt biomolecular structures on their own that can interact with proteins inside the cell. And we'll give a compelling example of that. This is how we get back to Down syndrome and Klinefelter syndrome. For completeness, I'll put the other functional non-coding RNAs. Here, SNRPs you've heard about in splicing and things. SNO-RNA is another one that's important for ribosomal RNA synthesis in the nucleolus. Question? Okay, so then if your question is ribosomal RNA sounds like a long non-coding RNA, ribosomal RNA. The definition of ribosomal RNA is that it's going directly into the organelle that is the ribosome, whereas long non-coding RNAs are much more general than that. I actually don't even 100% know how long the ribosomal RNAs are, even if they would fall within that definition. But if you say just loosely, yeah, if they're long enough and they're non-coding, you might argue that they're still in that same category. There's a much shorter history of link RNAs than there is ribosomal RNA. Okay, let's talk about microRNAs first. The biosynthesis and regulation of microRNAs is a deeply evolutionarily conserved process that goes all the way back to fruit flies. Plants have versions of this. And it was originally thought to be involved as a rudimentary antiviral mechanism in cells. What I want to talk about is the endogenous regulation of microRNAs in mammalian cells. There's 3 enzymes that I'd like you to be aware of, and we're going to start from the transcription of a microRNA locus from the genome. MicroRNAs are transcribed by Pol II, just like mRNAs, but they have this characteristic of having self-complementarity along a certain stretch of sequence. We talked about hairpins in other settings. Transcriptional termination is the one that comes to mind immediately. Here, there's complementarity on either side of this stem loop, such that when the RNA is transcribed, it's going to base pair with one another and form that hairpin. This transcribed hairpin that's capped, okay, just like any other Pol II RNA, then gets processed by an enzyme called a drosha. So what drosha does is clip off the ends and get this pre-microRNA hairpin ready for export out of the nucleus and into the cytoplasm where it operates. From there, this hairpin structure is processed further. The loop is cut off by an enzyme called a dicer. And these are self-explanatory. So dicer will mature the microRNA. And then the executor of microRNA function is another remarkable protein called argonaute, specifically, or AGO2, argonaute2, is what does the business end of RNA interference. Argonaute will take this microRNA duplex. There are 2 strands there. There's what's called the guide strand and the passenger strand. When argonaute recognizes the duplex, the passenger strand is kicked out and is degraded by exonucleases in the cell. The guide strand is then used as a template for base pairing against other RNAs inside the cell. if this RNA-loaded Argonaute 2 is called the RNA-induced silencing complex, or RISC. So RISC is the active RNAi machinery inside cells. When RISC encounters an RNA that is sufficiently complementary to that guide sequence, one of two things can happen. One, if the sequence is right and the binding is tight enough, Argonaute will then cut the RNA that was recognized by the guide strand. If you cut the RNA in two, now those two ends are degraded by exonucleases, and that RNA goes away. Conversely, if there's a more imperfect match between the guide strand and an RNA, Argonaute will bind but not cleave the RNA. But simply by binding and occupying the RNA, this can block protein translation, and then you can downregulate the protein product of that mRNA. So the microRNA machinery can downregulate or silence genes and proteins, either by degrading the mRNA or by blocking translation and preventing the protein product from occurring. This is very cool and very important for the lab. When I started doing research, messing around with the abundances of genes in mammalian cells was virtually impossible, to try to downregulate endogenous genes. So this was a windfall when it came out, because having a deep fundamental understanding of how microRNAs and RNA interference works enables us to engineer biomolecular reagents to then downregulate whatever genes we want inside cells. There are two ways, the field goes crazy, it settles down, and so I'm going to talk to you about the settle down way in which people do it. So you can exploit the endogenous microRNA and RNAi machinery in one of two ways. You can do it synthetically by synthesizing what's called a small interfering RNA, simply a duplex of RNA that is already the mature guide and passenger for a sequence of a gene that you want to target. There are rules about how you design those sequences so that they're very potent, databases that you can look up to find these. And you just synthesize it in the lab, anneal the two together, and then introduce that double-stranded RNA inside cells. You keep it to within 21 nucleotides. If it's too long, you get interferon type of responses. The cell thinks it's a virus. But if it's short, it will get loaded into the Argonaute complex and then target genes. That's the way you can do it synthetically. You can also do it genetically by encoding that hairpin-like sequence, either a microRNA-like sequence or a perfect match hairpin sequence, in a circular piece of DNA called a plasmid, or in a virus that integrates into cells. You engineer the genome of the cells to give them a source of a microRNA-like hairpin, which is transcribed off of a tRNA promoter, then processed by Drosha, Dicer, to get the duplex that feed back into Argonaute 2, and then you can downregulate genes. And so before, we talked about CRISPR and CRISPR-Cas9 as a mean to do knockouts. This is a mean to do knockdowns. If you have a very potent RNAi hairpin or duplex sequence, you can put it into cells and achieve 10-fold downregulation, 20-fold downregulation of an endogenous gene, and then do functional genetic studies from there. And all you need to know is the sequence. Questions on microRNAs? Yes?
This isn't about microRNA, but can you use CRISPR to do knockdowns?
Yes, so your question is going back to CRISPR and how do you do can't you do knockdowns with CRISPR as well? You can, and the way that you do that is by, there was the last slide in Professor Zunder's lecture about using nuclease deficient Cas9. So you don't use the end nuclease activity. You simply use the targeting specificity of Cas9 and then fuse it with a transcriptional repressor. So there are certain domains that may methylate a locus or put a whole bunch of repressive histone marks on a locus. You use the dCas9 to bring it down to a promoter of a gene that you want to downregulate, and then you shut down transcription of the gene. So it's Same idea, but at a totally different level of the central dogma, but using the different tools and technologies that we've been trying to get across.
So based on what they do, the small interfering RNA and single guide RNA, this would basically the same thing, which is binding to the right gene.
Was that the question?
Yes.
Okay, now I'm going to repeat that one because I was mid-drinking at the time. You're asking, is the single guide RNA for a Cas9 similarities and differences with the guide RNA for Argonaut 2? Similarities are that they're both using RNA sequence as a targeting mechanism for an enzymatic activity. Cas9 is a DNA endonuclease. This is a ribonuclease because you're cutting RNA rather than DNA. And it also has different recruitment partners. It exists in the cytoplasm. Oh, and another key one, we all have Argonauts in us. We have this machinery endogenously. Cas9 comes from archaea. So it's always, that's a synthetic mechanism that we put into mammalian cells. Those are the key differences. Good. Let's talk about link RNAs. Here's where I had that little bit of hesitation on the size. They say it's 200 nucleotides. That makes it long, but I wouldn't read into that too much. A couple other things. They're transcribed by Pol2. They're like any other expressed gene. They just don't have a coding sequence in them. So they're going to be capped. They can be spliced. They can be polyadenylated. But there's no open reading frame in there that is expressed. Because of that, they can do a whole bunch of things inside the cell. A lot of it is thought to relate to the base pairing, hairpin structures, and other types of secondary structure that RNA can occupy. And now you have a biomolecule with a complex organization that can be used as a surface or an adapter type of protein to bring different biological processes together. Here's one example of a very long non-coding RNA called hot air. And here are a couple of examples of the secondary structures that it can occupy, only over a couple of the domains of the hot air link RNA. I already mentioned that they can also compete with machinery like mRNA expression and microRNAs. We just talked about the microRNA machinery. If there was a long non-coding RNA that set up a whole bunch of decoy sites for another mRNA species, it can suck up all of the microRNAs and then prevent downregulation of the endogenous mRNA, as one example. And there are examples of these types of mechanisms in cells. And then they can also impact gene expression itself at the transcriptional level, and that is going to get us back to the syndromes that I started with. Because what we need to talk about is a specific link RNA that's related to gene dosage and a process called a dosage compensation, and another mechanism called X inactivation. And it all revolves around this long non-coding RNA called Xist. I framed the two syndromes as a gene dosage problem, right? Too much X, too much chromosome 21. All of us have, well, who has the gene dosage? Females have the gene dosage problem with the X chromosome. All the females have twice as many X chromosomes as males. And either we don't have enough or the females have too much. There are different strategies depending upon the organism for dealing with the gene dosage compensation on the sex chromosome. I'm going to talk about the strategy for mammals. So 2 X's, twice the copy number, twice the gene dosage. And when we talk about X-linked genes, those are genes that are on the sex chromosome, and these are the ones that are at issue with the gene dosage. So for mammals, the way in which the gene dosage of the sex chromosomes is handled is through a process called X inactivation. Early in development, this occurs in females. 2 X chromosomes. On a cell-by-cell basis, one of those 2 X chromosomes is inactivated randomly and permanently. And there's a name for it. It's called XI. All right, so the inactive X is the one X allele that is inactivated, and we'll talk about mechanistically how that inactivation occurs. The other one, the leftover one, if you will, remains active, and then all of the X-linked gene expression occurs off of XA, the active X allele. Now you fixed the gene dosage problem. So now females will only have one X gene dosage. The other one is inactive. And the mechanism of X inactivation depends critically on this long non-coding RNA, XIS, which stands for X inactive specific transcript. And XIS is a really remarkable link RNA. I have some things to say about it. The way in which it operates is by epigenetic, at least in part, by epigenetic modifications on the inactive X chromosome. What occurs is that XIS, when it is expressed, it will go back and bind to, it's called coding the X chromosome from whence it was transcribed. So it's getting transcribed and it's going back and binding onto the chromosome and coding it. And there are XIS binding sites along the chromosome that cause this compounding coding of XIS along the X chromosome, shown schematically here. So once you start transcribing, there are binding sites in the locus. It almost acts like a nucleating event for more XIS and then more XIS coding. So XIS can interact with DNA. That's what's shown here. It also can recruit proteins. So all of these protein complexes in the cartoon shown here, these are epigenetic modifiers that promote methylation of DNA, DNA methyltransferases, histone methyltransferases that promote histone inactivation, and then also HDACs, histone deacetylases, to promote histone deacetylation. So as XIS is being expressed, coding along that X chromosome that eventually will become XI, you're recruiting more and more epigenetic regulators to that chromosome and shutting down gene expression. At the end, XI forms an entirely heterochromatic chromosome and gets shuttled off into a cul-de-sac of the nucleus called a bar body. So you can do fluorescence imaging for XIS RNA. It's all coded along the chromosome. It's all condensed, and it's just in one little corner of the nucleus. And there it just sits forever. So as I had to do a lot of preparation for this lecture. So I was getting into this. I was very curious about this XI and X inactivation and the randomness of it. So this is a really cool, if you look into more of the mechanism. So I'm going to teach you math in the post-lecture on how the regulation of XIS and another molecule called T6 is sufficient to give rise to random and definitive activation or inactivation of 2X chromosomes. XIS itself and the mechanism that I just described to you is not sufficient to give rise to X inactivation. You need something else. And the something else is an antisense link RNA called T6. Why is it called T6? It's XIS backwards. And XIS is transcribed on the other, T6 is transcribed on the other strand of the locus where XIS is transcribed. That's how you get a perfectly complementary long non-coding RNA, because it's going off the other strand. It has its own promoter. And by virtue of this, you have competition between T6 and XIS. At the end of X inactivation, what you're left with is an inactive X where XIS is expressed and T6 is silent, or the active X allele where T6 is transcribed and XIS is inhibited. So I have about a 20-minute post-lecture to provide a mathematical derivation. I'm going to give you an introduction on dynamical systems theory from the perspective of XIS and T6, which explains how, in the language of differential equations, how these two competing processes and the interlinking of two positive feedbacks, or positive and negative feedback, can give rise to this split into two states of an active state and an inactive state. So now we have the explanation of Klinefelter versus Down. In Klinefelter syndrome, the X inactivation occurs on all X chromosomes except the one that remains active. So the extra X, if you have a third X, two X's and a Y in there, will get silenced, and the defects are relatively minimal. There's only a handful of genes that escape X inactivation, and they're the ones that ultimately give rise to the fertility defects. But with Down syndrome, you have no mechanism. There's no chromosome 21 inactivation. There's no X cyst there. And so there you have the gene dosage issue. And here you have all of the phenotypes associated with Down syndrome. So aberrant gene dosage on all the genes of chromosome 21. And chromosome 21, if you look of all the chromosomes, it's pretty tiny. Okay, and so that's even why you can have viable individuals. There are other examples of gene dosage issues, triple copies. One is in trisomy, so 3 copies of chromosome 18. This gives rise, that chromosome 18 is bigger than chromosome 21. It gives rise to a syndrome called Edwards syndrome. These defects are very severe. Again, I show them here. Many organs, and these children don't live past a year. Doing good for time. I'll say a couple additional things about X chromosome inactivation and X-linked disorders more broadly, because there's some other things, both in the disease setting and in the curious setting, that relate to X inactivation. So what does this mean for females? You are all mosaics. That in each of you, each one of your cells has selected one allele of the X to express and another. Tabby cats show this visually. It's a pigmentation locus of either an orange coat color or a black coat color. And so each one of these cells indicates which allele is active and the other one which has gone to XI. So the orange is XA here, and the black is the X allele here. If you're curious, the white, I looked this up because someone was going to ask me. That's another pigmentation locus, so that's kind of not related to X inactivation. X inactivation also features very prominently in hemophilia. Hemophilia is a sex-linked disorder strongly associated with males because the major clotting factors are X-linked genes. So it's overwhelmingly in males because if we get one busted copy of those clotting factors, we have clotting disorders. We can't compensate. Females, because they have two, even though if one of them is damaged or one of them is mutated and you can't clot, the second one is expressed in roughly 50% of your cells and can create enough of the clotting factor that there's no defect from there.
For the X chromosomes in females, let's say you got one stroke, like a clotting stroke on one, but you got a different stroke on another. Can you inactivate parts of one and keep parts of the other chromosome active?
Yeah.
Or it's just you have to pick one?
So the question is, I'll rephrase it a little bit. Are there any opportunities for partial X inactivation where you may have some on one locus and other on the other? No, not by the mechanism, because the mechanism of Xist is right local on where it's being transcribed and it's self-reinforcing. So more if you want to think about it from an evolutionary standpoint, you need it to be definitive. And so it's more what the pressures have been is to totally shut one off and leave the other one untouched so that you can maintain the gene dosage. But I'll talk about one example of a variation on that theme to close out. From the two X copies here, from the female perspective, are good from hemophilia, but they're also very tragic in other disorders. So Rett syndrome is another X-linked mutation for actually an epigenetic regulating protein called MECP2. This is a protein that recognizes methylated CpG sites in the genome. And Rett syndrome patients, this is a mutation not in the germline, so it's not an inherited disorder, but it's one that picks up during that spermatogenesis or oogenesis that I talked about before. So the MECP is on the paternal side, and if it's inactive, it's defective. What happens is that when it comes up, males with only one copy, if they pick up that one copy of broken MECP2, it's not compatible with with life. And so you have no males with this because they don't survive in utero. But females, because there's the second copy that can compensate, and you have this X inactivation. So what happens is that some of the cells will have the broken MECP2, others will not. And so the good copies allow the children to look normal at birth. This is the same individual, but as they age, they go into their early teens, it gets to be this progressive neurodegenerative disorder. And there's people actively working on trying to find ways to study this, but this is a consequence of X inactivation and the reason why it's a female-specific disorder. Yes?
For like the cat, it's like obviously it's grouped together, like the cells that are inactivated in a certain chromosome or not. How does that work? Like how do the border lines work?
Yeah, excellent question. I'm glad you asked that one because I said, oh, it's random, right? The choice, the X inactivation choice. Then why do you see these fields of one color versus another? The choice for X inactivation happens rather early in development. So what you're seeing is the choice of the progenitor that then expanded and differentiated all across that field of pigmentation. So as those pigmented cells in the dermis of the cat, as an example, differentiated, there was a choice for X inactivation, and then they gave rise to all of the skin cells in that field. And then they all merged together and things like that. All right, last thing to bring XIS back together with Down syndrome. I think, Nicole, you had a question about this before, if I didn't forget. Is there any ideas of ways in which you could use genetic engineering for something like Down syndrome when you have a gene dosage defect? There was a proof of concept on this for Down syndrome. I'll talk about a little bit more technology because this one was kind of a crazy experiment. What these investigators did was take a skin biopsy from a Down syndrome patient, fibroblasts. Professor Barker talked about fibroblasts. Yes? Okay, so it's important for scarring, ECM, but there's plenty in your skin. Okay, so you take these fibroblasts, and then what these investigators did is genetically engineered them with transcription factors that collectively reset the epigenetic state of the fibroblasts and allow them to go back in time to become what's called an induced pluripotent stem cell. So these are the original, they are very close to the original embryonic stem cells that give rise to all the differentiated tissues in an organism. So they took these cells back in time epigenetically, reset all the marks, methylation, histone marks, et cetera, made an induced pluripotent cell, or iPSC, and then genetically engineered the trisomic 21st chromosome, one of them, and inserted the XIS locus, the long non-coding RNA. The way that they did that, the ZFN, those are zinc finger nucleases. Think of those as old and very inconvenient ways of doing what Cas9 can do now. So they can cut in a sequence-specific manner, but they're a total pain to work with. But this is what they had at the time. So what they did was cut the 21st chromosome, one of them, and put in the XIS locus. And then what they could do, they engineered it so that it was inducible with a small molecule called doxycycline. They turned on XIS expression in the cells, and what they wanted to see was if you turned on XIS expression that was engineered in the 21st chromosome, could you coat that chromosome with XIS and put it into a bar body and shut it down, just the same way ordinarily happens with X inactivation. And it looks like it worked. So there's the XIS, bar body, all the functional studies and things, being able to reverse the gene dosage. So at least in a proof of concept, it shows a nice example of how one could combine the concepts of X inactivation, long non-coding RNAs, epigenetics for developmental syndromes. If you think all this is crazy and cool, I will direct you to Nathan, who's our DNA methylation guy, Sanchita, who is interested in X inactivation and Rett syndrome, like I touched on, and then a Ninja, who's the head of their department, is a big guy in non-coding RNAs. 2 minutes for questions. Yes? Yeah, so your question is that when the X inactivation choice has occurred, what happens then if that cell is still replicating? Yes, it is. So once the choice is made, it ends up being self-reinforcing in all other DNA replication events. That gets back to the question why you'd have that field effect. There's only a limited window of indecision on which X allele is chosen, and then once it's chosen, it's permanently inheritable from that point forward. That, again, is an there's an epigenetic character of that. Yes, it's still replicated. It doesn't disappear. It's just once the choice has been made, that's XI all the way through.
You said like the inactivation happens at early development. What would that be? Like, are we talking about in the womb? Are we talking about out of the cell?
Yeah, you got me. I can't say the exact stage, but I don't know. Yeah, I would say definitely in the womb, but I can't tell you at how far along. I would, if I have to guess, like fetus level developmental stage, but I don't know the answer to that. All right, have a nice weekend. I'll see you on Tuesday.